Everybody. Hello, hello. So, I am going to do the review. I'm going to be shutting off the camera and I will just narrate it to you. For those that have not had me before, when I do a review, and I may have to do this in two sections because I'm going to probably have to leave here from home. But, and I'll send you the second half. So, we'll get done what we can. That's there. So I will proceed to do the review starting now. And the way I do it is I look at the exam. I mean, you have the study guide and materials, and if you have any questions, just ask me in class. Um, and here we go. So basically, I would think the chapter that you see the most material of is certainly the what Cowan's chapter 4 and Nestor's chapter 3, basically the architecture, the anatomy of the cell, the gram positives, gram negatives, cell walls, endospores, etc., that type of stuff. But we have some material from chapter 1, and perhaps that's the least of the material. And then the greater bulk of it is from, and then the rest of it is from that chapter on the physiology, the growth, and temperature levels, the bacterial growth curve, etc. So we start with the introductory chapter, as we always do, and most importantly is Pasteur. And Pasteur was all about refuting the theory of spontaneous generation. So his idea was germ theory, these things we can't see in the air. That was what that famous experiment with the goose nest flask was all about. Okay, so that's really where his area of expertise was. Remember, Koch did anthrax principally, which was troubling the farm animals. A lot of the previous theory was called spontaneous generation. Fancy names are abiogenesis for spontaneous generation, and then what we know is true today. Basically, what sort of generically was called uh, germ theory. And again, Pasteur's area of expertise was in looking at these microorganisms like yeast and addressing them with regard to the spoilage of wine, which really gave him all the credibility. So, and again, the Pasteur and Koch were really surely the two most major microbiologists of their era. They basically, and Koch was the lab guy. He was a little bit younger and lived a little bit longer than Pasteur. And he basically cultivated things and all the things in the lab: staphylococcus, tuberculosis, anthrax, grew in the lab, developed postulates to be able to do that, developed agar, plating techniques, all the stuff you do in lab. Koch did. Well, Pasteur was more trying to find the answer to the problem rather than the science behind the problem. That he wasn't scientific, but I mean, we're starting to utilize different kinds of serum in an effort to try to treat people with rabies, which is really what we do today by injecting antibodies, or again, finding out about what was causing the diseases, verifying this, finding the wine industry, really starting immunizations, coining the term virus. So he was really, from a theoretical standpoint, something special. And a lot of this, eventually, the beauty of microbes is what they've opened the doors for us. Being able to understand that since they do business differently, there are lots of materials we find in them, enzymes and chemicals in them, that are able to. Now we're using an enzyme that we find in, of all viruses, HIV, to do what's called gene therapy, to sort of uh, retrofit, and as we call it, a retrovirus, because we can write a stream of RNA and transfer it to DNA and try to get it incorporated in our cells to effectively reprogram them, resequence them. And we think of a thing like this. And we do the same thing with yeast and E. coli. We resequence their DNA because they reproduce so rapidly and with so little cost that we can, and that's what genetic engineering is. We can create in the laboratory now, because we have enzymes that can chop up DNA that come from bacteria called restriction enzymes. We're able to analyze the DNA and then create sequences of the DNA and make our own little strands of DNA, and then those little strands can be inserted by way of plasmids into susceptible organisms, easy to grow, like E. coli and yeast, and we can make things, starting with human insulin. They're there. But the problem is we haven't understood what viruses and bacteria really were. So bacteria are a living population. And when you treat a living population aggressively with materials to kill it, effectively it's chemical warfare. That's what the problem with antibiotics were. And what happens, they respond by evolving. And this descent with modifications, Darwin and his theory. And what happened was we've developed mutations. So emergent diseases are mutating forms, diseases we find in areas we're going into, changes associated with the changing nature of the population, response to drugs, and things like that. These are all things. So, our biggest problem we deal with in the form of emergent diseases is finding new ones because we expand our population and go into areas. And most importantly, for sure, mutations. And then viruses, which mutate anyway, much more rapidly. And we do the viral, you'll learn about RNA viruses like COVID, like influenza, okay, like RSV that mutate. And so that's going to be the talking points for the viral, which is part of this second unit that we have just started now with methods of control. And then we're going to carry on with genetics. And then finally, with viruses, that's the second big unit of micro that I do. What are resurgent diseases? Diseases we thought we had eradicated. And there's four major reasons. Okay, and really tuberculosis and to a lesser extent measles are the reason. Vaccine laxity, that accounts for measles. For tuberculosis, more people are immune suppressed because we treat immunosuppressive diseases, and we found a lot of immunosuppressive disorders, not the least of which was HIV. And so because of that, and because of the availability of treatment for certain things, people became very lax in how, whether doing vaccines or being subjected to different types of treatments electively. Taking an immunosuppressive agent is not uncommon at all. I take one for the kind of arthritis I have. And you have to make sure you're tested to make sure you don't have TB to take that because you would be predisposed to it. We have a lot of people living longer. That's part of it. And the fact that we travel all over, we may go to an area where we didn't have a disease and bring it somewhere else. So worldwide travel plays a role. So all of those are really the sort of the backstory for the resurgent nature. So we have both emergent and resurgent, and they're not the same. Resurgent means they're making a comeback, and we thought we have eradicated, and emergent are different. There were other founders in microbiology. Semmelweis, looking at those studies even before Pasteur about dirty hands and soil things and delivering babies in puerperal sepsis. Lister, following following what Pasteur talked about, and then adding all those antiseptic ideas that we call them now with phenolic compounds, by washing instrument, washing hands, cleansing skin. Those are all really important. And if you want to go back to Van Leeuwenhoek in looking at the microscope, that's fine too. Those are all people that played a role. The guy Reddy with looking at decaying meat is an important player as well. So there's been a lot of foundation for this, and it just keeps going on and on. Okay, that we begin to see, and we don't really touch so much on the modern era here, but you will, depending on where you're going, you'll learn a little bit more about advanced DNA testing, and even though we use it commonly, if you do Ancestry or 23andMe or whatever is popular now, and a lot of people are into it, I'll believe me, a lot of the faculty here, they can't stop talking about it. So I personally have not done it. I'm not that curious. Okay. And then we get into really what the constituents are of bacteria. Well, bacteria, number one, hit parade. And remember, the word unique means it's only in that species or in that organism, or that's a particular trait that we just don't find. So what's unique? Peptidoglycan. All bacteria have it. Lipid A. Okay, that's unique to the gram-negative outer membrane. The only bacteria that has an outer membrane is the gram-negative, and the lipid A is in there. And obviously, it causes problems because it's unique. We're not used to it whatsoever, so it's inherently toxic to us. We have to have a reaction to it. So that's really when you talk about unique characteristics or distinguishing character. Unique is a very, very specific word. For instance, of the eukaryotic microbes, what makes algae unique? They're the only ones with chlorophyll. So that makes them unique. If you want to think of fungi, they're the ones that are of the eukaryotic microbes that are saprobes, that they eat dead stuff. They're fancy terms you don't have to know called exodigesters. They secrete enzymes, break down the stuff, and then basically use that as nutrition. So, I mean, algae have a cell wall, bacteria have a cell wall. That's not unique. But algae have chlorophyll. They're the only ones that are photosynthetic. Okay. Those are distinguishing traits. Bacteria and archaea are very similar size-wise, and they have cell walls. But the trouble is the cell wall doesn't have peptidoglycan and archaea, so it's a significant difference that's there. Size, they're about the same, and the other big difference is where they live. Archaea live only in extreme environments. Bacteria can live there, but archaea can only live there. So that's the interesting part that's associated with about that. When it comes to things like viruses, they don't play favorites. A virus is really just a little nugget surrounded by protein of genetic material, either DNA or RNA, but never both. Because if it had both, then it quite possibly could be living. Not the case. And they can infect all domains. The bacteria can get a virus, and we're going to see that in genetics. Archaea can get a virus, and you and I obviously can get a virus. And so it doesn't matter that viruses don't play favorites, and viruses inherently are going to cause some type of damage. Maybe it'll be a modification that's useful in some instance to an organism, but for the most part, it's not. Okay. So I think that's important. And prions are the odder still because they don't even have any genetic material. They're just a misfolded protein. So they're non-living in their proteins, and they're misfolded, and all they do is damage other proteins as much as we know about them. And as you probably have discerned by now, that's the group that we know the least about. So I just wanted to keep going on that sort of thing, so you kind of know where we're heading. Every so often I have to check back to get my place and make sure I've got everything in order. So things that are really focal points. Endospores. Endospores. Remember, they're in the spore or endospore stage. Bacteria came originally, we thought, from plants, even though that was wrong. So when you hear the term vegetating, that's a bacteria that's growing in the lab. Growing on those culture plates are vegetative bacteria. Are there some endospores? Sure. If you're using an endospore form, it usually comes from either the ones we use are from bacillus, but they also come from Clostridia. And there's some oddball ones, one called Sarcina, which we really don't utilize, that are there. They effectively are indestructible. Okay. They have a forespore that eventually becomes the endospore itself. Okay. The spore coat is reinforced with peptidoglycan proteins, that dipicolinic acid that comes in and surrounds it after the mother cell degrades. So you have all of those types of things that are there. Calcium comes in to harden the shell, sometimes has an additional protein coat. And it's quite remarkable with regard to that. It takes a few hours for them to form. They're not something that necessarily happens rapidly. And sometimes you'll see them within, and we can use that for identification. Sometimes they pop out. And that's when you do the endospore stage, you're going to probably see some of that. Maybe that's something you'll be doing this week or next week. So we have different shapes that are important. They basically are three forms. They can either be spherical, which we call cocci, and they appear round because we're looking at two-dimensionally. We have rod shapes, which are called bacilli. That's with the small b. The big B is the one that's the genus. And then we have various kinds of spiral forms, spirilla, spirochetes, vibrio, and some irregular forms that go along with that. Again, the ones that are pleomorphic are the ones that don't have a well-developed cell wall, even though they have some remnant of peptidoglycan, and the most famous of this is mycoplasma. Sometimes they have a very thick capsule. Okay, that's that glycocalyx. When it's thick and well organized, it's a capsule. When it's sort of random and non-specific and not highly organized or thick, inherently it's very sticky because really ultimately they're sugars. Okay, when we see those sugars become more like long-chain fatty acids, that's when they tend to be more encapsulated. So it depends what's attached to them. But when it's just the sugars themselves, they form that slime layer or biofilm. And the biofilms have a lot of relevance today, both in a destructive sense and in a utilizable sense. So there's a lot that goes along with that. They all are living things, bacteria, so they all have to have a semi-permeable membrane, phospholipid bilayer, embedded proteins that can, to some extent, move around effectively. And by definition, those proteins and the nature of the membrane allow some things in and some things don't come in. So it's semi permeable by all of it. Does it have to have cholesterol? No, bacteria don't have cholesterol in that. That's there. Do they do energy transformations in that? Not always. Okay, because some organisms don't do energy transformations in that regard. So all of those types of things that are there. The key is selective permeability. That's what a living membrane is all about. And when it comes to movement across membranes, really the big player continues to be, just like it is in our cells, concentration gradient. That's the big influencing factor on things like simple diffusion and facilitated diffusion. And bacteria tend to conserve their relatively limited energy resources. Bacteria have very specific ribosomes based on that density. You have a small and a large subunit. They come together. It's a 30 and a 50, but when they come together, rather than just adding them, the density changes or alters and they become what we call 70S units. Where in our eukaryotic cells, our ribosomes are 80S, with the exception, and that's that critical piece about endosymbiotic theory. Without that critical piece or understanding that within our mitochondria in our cells, or chloroplasts of mitochondria in plant cells, they have the same, they're basically bacteria that were ingested and became utilized by some primordial early cell billions of years ago. And now they have become organelles that can replicate on their own. They have their own membrane. They're membrane bound organelles. That's why they're part of eukaryotic cells. And that's a big distinction. It's really important in antibiotics because a lot of them target 70S ribosomes to address bacteria. Most famously, the Z-Pak, and everybody knows about that. Everybody knows that somebody's been on, knows somebody, if not themselves, been on a Z-Pak. We'll learn about the specifics about why that's such a valuable tool. Okay, but it targets the 70S ribosome. So the difficulty with that is side effects, because we have some 70S ribosomes in our cells that are there. The peptidoglycan itself is important. The alternating NAMs and NAGs, you don't have to know the name. The fact that the glycan part is really the bricks, and the peptide interbridges and tetrapeptide groups are more of the mortar. That is very thick in gram positives, 30 to 40 layers thick. In gram negatives, you have a layer or two. In gram negatives, it's sandwiched between the inner and outer membrane that are there. The tetrapeptides and the peptide interbridges allow it to be cross-linked. Okay, the nature of glycans is nitrogenous carbohydrates. The big distinction is that there's differing amounts in different kinds of bacteria, particularly gram positive and gram negative. The gram negative have that interesting outer membrane. They have a regular inner membrane. Then they have a layer or two of peptidoglycan surrounded by an interesting material called periplasm, where it has a lot of activity comparatively, bringing things in and out, being able to defense mechanism against antibiotics is kind of interesting in that regard. And then this outer membrane, whose outer leaflet, the inner leaflet's phospholipid, the outer leaflet is this lipopolysaccharide, and that's the odd one. That's the unique feature, the lipid A, the outer leaflet of the outer membrane, the fatty component. That's something that's only seen in gram negatives, and it's so foreign to our cells that we immediately respond to it, which by definition makes it a toxin. The response is always by our defensive system. And if it's in sufficient amount, it becomes a very, almost like an allergic reaction, if you want to think of it this way, that way. It leads to something called endotoxic shock that we'll talk about. And what that means is you have a large outpouring of immunological materials. They dilate blood vessels, they lower blood pressure, accelerates heart rate, diverts blood flow from vital organs. That's what shock is, and shock is potentially deadly. So you have to treat that very, very aggressively. Classic example, ruptured appendix would be an example. That would probably, if you wanted, without looking into cancers and things like that, a ruptured appendix would be the definition of that. Occurs. That's that very famous lipopolysaccharide layer. It's only in the gram negative outer membrane. By definition, the lipid A is an endotoxin. Endo means it's part of the component package. It's there, it's a structural component. Exotoxins are toxins that are spit out, and they're spit out by both gram positives and gram negative cells. Toxins is how bacteria do most of their damage. Viruses, as you will learn, do it differently. They invade and damage cells, and it's that cellular damage or the immunological response to them that really creates the problem. It's mostly the immunological response, like COVID would be the example of that. That O specific antigen or O specific polysaccharide is a sugar chain that's unique in the sense it's an identifier. And that way we can categorize, and that's really important because that tells us what the toxicity package is to that bacteria. Is it a killer strain of E. coli or is it regular E. coli? Is it a killer strain of salmonella or is it a regular strain of salmonella? Because once you've identified and typed that, it gives you some insight in how to go about treating. So that's what identification plus identification is used to track it, particularly in the sense when we get to the epidemiology components of the course, the disease component. We don't focus a whole lot on that, but you do need to know some terminology about epidemiology. And most of us became, unfortunately, very familiar with epidemiological terms like pandemic, which is an epidemiological term, obviously during COVID. Because it has membranes inside and outside, you got to have openings, pores, for lack of a better, protein passageways. So they can be pores or porin proteins that are there in the gram negative cell. You don't need that in the gram positive cell because peptidoglycan is porous. It's naturally the water can get through it. It's a different kind of a structural basis. So once again, just to review that and just to reiterate, the glycocalyx are proteins with sugars attached to them on the exterior of all cells, ours included. Okay, and some bacteria commonly form slime layers, and if it's thicker and more organized, capsules. And again, we utilize those. Sometimes they contribute to their activity. The capsules are very difficult for our immune system to deal with because effectively we gobble up invaders when they're there. Then we have the whole issue, and a lot of my focus points between eukaryotic and prokaryotic cells. So a lot of it'll say in prokaryote or in eukaryote, in bacteria or in our cells. So look for that in any type of the questions that are there. So we have a sperm cell, men do, with a flagella. It's totally different than the flagella that's typically found in most bacteria. We use the flagella as a way they move around. Our sperm cell's flagella is an internal protein appendage. It's surrounded by membrane. It's just a chain of protein made of microtubules that whip back and forth and move that sperm cell along its journey. In bacteria, they're for movement too, but it's like a hair that protrudes. It's not bound in membrane because that's really the definition of what eukarya versus prokarya are. Prokarya just have an outer membrane, and so it's like a hair that's protruding. And we use it more for identification, and the more they have of those, the more mobile, or what we call motility, which is something you probably will study in the laboratory. They're made of a protein that's not like microtubules at all, and obviously size-wise, they're immensely smaller. They're extraordinarily, like 1,000 times the size of a sperm cell flagella would be, as compared to maybe a tiny flagellar appendage in a bacteria, 100 times surely, maybe as much as 1,000. They're made of something called flagellin. In our cells, the flagella have that classic 9+2 arrangement. So does cilia. We don't find cilia in bacteria. They have various sizes of protein appendages, smaller than a flagella and made of a slightly different protein, okay, called pilin or pili, okay, but they're hollow. So they're very, very tiny, kind of a little bit like a microtubule, but extraordinarily smaller and a totally different protein. No 9+2 arrangement, but there's a hollow kind of an arrangement that it has associated with it. And that's the one that's used, as you will see when we get to the genetics unit, to transfer or as a conduit or passageway in something called conjugation for genetic material to pass from one bacteria to another. And it's not a form of reproduction, as you will see. Okay, that's why it was misnamed, but they thought it was, but it's not. Bacteria are totally asexual in their reproduction. And then tinier still, and it's probably made out of something like pylon, though it doesn't have a specific name, are fimbriae. They're like little bristles that are coming off, and very much like bristles, they attach. So they're kind of interesting in that regard. So that interesting pilus is known as the sex or F, really more often the F pilus or fertility pilus, because it's all about being hollow and allowing DNA material to be transferred through. It's shorter than a flagella, and again, it's made out of pylon. It has nothing to do with microtubules. A lot of bacteria, and really this is what goes in a pilus when it's transferring, is part of a plasmid. Sometimes the whole plasmid, sometimes only part of the plasmid, depending on the situation. And plasmids are single circles, small circles of DNA, maybe 25 to 50 at most genetic locations that are in there, actual genes that are there. They form a circle. That's what we genetically engineer. We splice, because they're small, we can splice DNA into the circle at the right place. And you can use that because they replicate along with the cell once they're incorporated. That's the beauty of them. Most bacteria have them. Most bacteria have more than one. But understand, they're non essential. They're useful. They typically have things that can help the bacteria live longer, okay, reproduce more easily, particularly resisting antibiotics. That's where we find most antibiotic resistant being moved by way of plasmids. That's the helpful thing to bacteria, but they can disappear as well. So they're useful but not essential. That are there, and again, there are more than one. And when we get to how conjugation occurs, in a matter of minutes, literally, that bacteria can change, and that so it can be a very very rapid process that's there. So again, underscore the differences between what the components are, whether they're microtubules or microfilaments or intermediate filaments, things that we classically see, though we do see some microfilaments in bacteria. Mostly we see those in abundance in eukaryotic cells, membrane-bound organelles, ADS ribosomes, pairs of chromosomes, etc. So all of that's important. Review that endospore animation. Again, the endospore, okay, starts with a 4-spore, the peptidoglycan. It involves a couple of hours. Okay. It involves dehydration because water is extruded. It involves calcification because calcium comes in. And what occasions it more often than anything else is a lack of nutrients. So how do you read that? You read that as decreasing amounts of carbon and nitrogen compounds. Water also would play a role, but I think it's an ancillary or a role, an assistive role rather than the important role. When we look at eukaryotic cells, bacteria have a cell wall. They don't need stability. Eukarya, particularly protozoans, do not have a cell wall per se. And what they utilize is agents. And even algae and fungi, all eukaryotic cells utilize different kinds of sterols. Sterol means they're derived from cholesterol with various chemical substitutions that are made inside the cell. Maybe the best known, cholesterol is in animal cells like ours. Ergosterol, ERGO, sterol, all one word, is the one we find in fungi. That's a very useful target for antifungals, and almost all the antifungals will be used, derived from attacking the ergosterol. And we get to that in the third major unit that we do, really in the antibiotics unit. Bacteria have a very scant cytoskeleton with a little bit of microfilaments. They do not have intermediate fibers. They do not have microtubules at all. Those are strictly in the realm of eukaryotic cells. And then the usual stuff about eukaryotic cells, membrane-bound organelles, nucleus, nucleolus, usually one or more. They do mitosis. Nucleolus is where ribosomal RNA is generated. Mostly they do mitosis, particularly in microorganisms. Some do meiosis, mitochondria and chloroplasts. Chloroplasts, if they're more in the plant family or in the algae family, those are all do. That's where energy transformations occur in eukarya. So and that's stuff that you should be amply familiar with. And then, of course, we get into more of the physiology with it, the different air categories. You and I, are obligate aerobes. We need to have oxygen. Some bacteria are like that. They're aerobic bacteria. They had the absence of oxygen would kill them. Okay. By the same token, there's obligate anaerobes. Most famous, all those Clostridia, tetanus, and botulism, and C. diff. They don't. Any oxygen would kill them. They don't have the means to defend themselves. They don't have the superoxide dismutase and catalase, or they don't have one or the other or both, or they don't have an adequate amount. But obligate anaerobes don't make any of it. Okay. So they're destroyed by oxygen. They very often we use oxygen to treat them. They grow in dead stuff. They grow deep in the soil. They flourish when there's no oxygen that can get through to them. So they're very much different. Then you have obligate fermenters. Okay. Obligate fermenters are they don't even have an electron transport mechanism. They don't rely on oxygen either. Oxygen is a source for the electrons to go to because they don't have an electron transport chain, or nitrogen or sulfur, which is what the obligate anaerobes utilize as an acceptor for electrons. So they just ferment. There's not a lot of energy to do, but so they do because they're exposed to oxygen. They can grow in an oxygen environment. They do have some amount of those agents, either catalase or superoxide dismutase, to be able to function. In the lab, we utilize what are called facultative anaerobes. Again, facultative is an important word. It means it has the ability to grow relatively easily. So it'll utilize oxygen if it's there. If it's not, then they can ferment. So the E. coli that you use in the lab, that's a facultative anaerobe. Staphylococcus, because we have an aerobic lab, grows fairly well. Streptococcus basically undergoes that is in that category of facultative anaerobes. It has enough mechanisms to deal with oxygen, but not a lot comparatively. So facultative, they're able to grow in the presence of oxygen. They're able to ferment in the absence of it. Okay. But the absence of oxygen, even for a short period of time, isn't going to destroy them. Okay. So all of that is important. So you should remember, just to reiterate, know the layout of the cells, peptidoglycan, bacteria, and gram-positive outside the wall, and gram-negative. When you go from outside the wall, you have the outer membrane, then the peptidoglycan and the periplasm, then the inner membrane, then you get into the cytoplasm and into the sort of the interior of the cell. So all of those are part of it that are there. The temperature areas, the ones to know, are really only three of them: mesophiles, thermophiles, and psychrotrophs. The psychrotrophs, they're the ones you see in the refrigerator. So they're there for maybe 4 to 5 degrees. So typically that's about the temperature of a fridge, to about 20 degrees Celsius. The thermophiles, about 45 to 70, and between 20 and 45 are the mesophiles. Those are the ones that affect us the most. So in the refrigerator, basically, how do you reduce spoilage? You're slowing down the growth process of those organisms that cause spoilage, because most of them, frankly, are mesophiles, okay, that are there. So that's all the relevant stuff that we see with regards to this. Oh, one more, the microaerophilic ones. Microaerophilic bacteria have been associated with, and there's three different diseases, so they're significant. Okay. We know there's an association with gastrointestinal ulcers. I mean, I don't know if I buy that completely, but association would be a fair way to describe it. So they may contribute to the cause. That's the Helicobacter pylori. They're definitely microaerophilic Lyme disease and syphilis, which are both spirochetes that are there. And then the other one that's interesting is that flesh-eating bacteria that you hear so much about. There's a number of types of it, but the main one is a variation of, interestingly, the strep throat strep that happens to be on the skin, and under the right kind of conditions, generally, when someone gets a cut, and it's almost classically you hear about somebody with a needle being jabbed into them. We were going to look at it called necrotizing fasciitis. They're out fishing, they get their finger caught in a fish hook because a lot of times we'll see them associated with water where that bacteria comes in. So there's kind of nuances that are there. So they are particularly nasty. And we're going to run into this because we do a unit when we look at the diseases of wounds. And classically, you would consider that mostly a wound disease, maybe with the exception of syphilis and Lyme disease, because they involve they're transmitted differently, and we'll get into all of that. So when you hear about toxic derivatives of oxygen, they are the fact that we periodically make molecules. Anything that utilizes or exposed to oxygen has to have a defense against it. And so that's when you hear about things, because they're normal metabolic processes of oxygen. Okay. They're the leading cause of modern cancer theory. How do we fight them? We utilize superoxide dismutase and catalase in our cells. Our cells that have them are called peroxisomes. The only category that's really relevant with regard to pH are neutrophils. Okay. They're typically pH 6 to 8. They can be as low as 5 to 8 that are there. Understand what happens when we have prokaryotes that, like bacteria, when they're in a highly salted environment, they undergo plasmolysis. That's that classic experiment. That's there. It's just being in pure water because the cell wall doesn't bother them. But it's the water being pulled out of the cell and bringing the envelope away from the cell wall that creates such a quick problem. So our cells are similar to most bacteria that we encounter. So we're chemotrophs or organochemo heterotrophs. We eat all types of different organic chemicals to produce energy and make building blocks. Where plants are autotrophs, and typically they put together their carbon compounds utilizing photosynthesis, which effectively is carbon fixation, taking carbon dioxide, inorganic, out of the air and affixing it to, like microbes do nitrogen fixation. They nitrogenate carbon compounds. They're there. So microbes do both carbon fixation and nitrogen fixation. And again, if it doesn't require a lot of growth factors, facultative, grows easily. If it requires a lot of growth factors, fastidious. That's when you're going to use what are called enrichment augers in the lab. And strep would be a classic example of that. Strep does not grow well on most augers. You have to enrich it, and you typically use sheep's blood to do that. Don't forget the CHONPS acronym, the way that bacteria move around. They're either attracted or repelled. It's that run, tumble, run because they have limited energy resources. Don't forget that. All the different attractants, chemical, magnetic, light, air, all of those things are part of it. There's a lot of different things that attract, even temperature. Some bacteria like it cooler, some like it warmer. And then the growth curve. 4 phases. Lag phase, basically adjusting to the environment, limited reproduction, hard to measure. Anytime bacteria goes in a new environment, and remember these are closed systems, then once it ramps up, it goes into log or exponential phase. All it does is reproduce by binary fission at an extraordinary rate. All the cells are growing, and it will do so as long as there's adequate nutrients and no problem with waste products accumulating. If they produce primary metabolites then, which are where we find useful agents like with genetic engineering, and absolutely the best time to attack them with antibiotics because they'll gobble up anything in their environment. So they're very susceptible. They become less susceptible as conditions change, when there's less nutrients and when there's an accumulation of waste products. They begin to mutate and the growth slows down and they lead to stationary phase where there are as many growing as there are dying. So the net growth is 0. Now you're starting to develop resistant forms, and what do they survive on? The cells that have died explode and they gobble up their nutrients. They gobble up their components as for their own sustenance that are there. That's when we see a change. They produce secondary metabolites. That's where they produce antibiotics because they want to destroy susceptible organisms so they can survive. And also, if they can form endospores, they do it then. And so we can genetically address these in the laboratory by something called continuous cultures or something known as a chemostat. And that's what big pharmaceutical companies do when they want to make antibiotics or they want to make human insulin. They can either keep it in log phase or in stationary phase, whatever works. And death phase just means more dying than are growing. Okay. And that's because sooner or later school's out, and the cells are going to, it just isn't as fast as growth phase, the acceleration. Remember, it's a logarithmic curve. That's part of it. So, other things that are noteworthy as we get to the end of this relatively long discussion. The mesophiles are by far and away the most common. They can be in gram positives, gram negatives, different kinds of forms. It doesn't matter. You typically in micro you don't paint with such a broad brush. That are things like that. So their reproduction is asexual. Eukaryotic microbes are mostly asexual, but mitosis, but some can undergo meiosis as well. The distinction between the kind of ribosomes is important. We only find effectively all of the ribosomes in bacteria are free ribosomes that are there. The nature of the membrane, the unique one being the lipopolysaccharide only in the gram negative, particularly that lipid A layer being the real problem. And understand endospores are really not a form of reproduction. They just sort of flip flop if they have that capability when times are adverse, and then they go back to the vegetative stage that are there. The distinction, again, living things have both DNA and RNA. That's why viruses and certainly prions, which don't have either, are part of it. You should know some examples of typical emergent diseases, ones you've heard about recently. Most of the time, they're associated with mutations, and the resurgent ones like tuberculosis and measles. Okay, remember about exotoxins, that's important. And I think that's about it. So thank you very much. We managed to get everything in without me being interrupted, which is shocking. And I will be talking to you tomorrow, no doubt. Thank you. Bye-bye.